Hi, everyone. It's my distinct pleasure to introduce here Andrei Levchenko of Yale University. Andrei is, without a doubt, one of the foundational figures in systems biology. This is one of the topics of our conference, of course. So he has graduated a while back from Columbia University with a degree in biomedical engineering, and since then contributed to many, many different aspects of systems biology, starting with his now classic papers on modeling of NF-κB circuits, which was probably one of the first modeling papers in the system biology and the one to which most modeling work in that system is compared to right now. He went on to introduce a couple of additional mathematical models, like, for example, the Legge model for gradient sensing in spatially extended cells. He explained the role of scaffolding in signaling, and then after that he decided he's going to do experiments as well, and there he became one of the very few people who actually are equally well known for his mathematical skills and contributions and experimental contributions. He introduced things like nanopatterning to study chemomechanical signaling and chemomechanical effects in cells, and at the same time he additionally worked out things like information theoretic channel capacities of various signaling circuits and many, many other interesting questions. And so with that, oh yes, and one other thing, a couple of years back he moved to Yale, as I said, and he's now the director of the founding director of the new Systems Biology Institute there, where they're doing wonderful thing. I encourage you to go to their website and see what it is that they're doing. And so with that, here is Andrei.
Thanks a lot, Ilya. Thanks to the organizers for inviting me, and thanks to the great hosts that we have here and the great city of Atlanta. What I'd like to do is to say that I'm not a mathematical biologist per se. As Ilya mentioned, we have complete inability to focus on anything, and so we do a lot of things. So I'm going to try to talk about a couple of things, but more importantly, perhaps, to think about questions that might be interesting to you, questions that have been emerging over the last maybe decade or maybe last few years, and try to illustrate that using a couple of examples. Before I go into that, I'd like to thank people who actually have done the work. I'm just the cheerleader, trying to sort of cheer them on, particularly the talented graduate students and postdocs in my lab, and the three of them are mentioned up there, and the great collaborator Sasha Popel, who is a mathematical biologist and also the funding. So I guess we all come into the field hoping to resolve wonderful, grandiose questions and use tools that we know how to use. And mathematics is a wonderful tool, a universal tool. And the good news is that biology indeed can be described with mathematics. We actually have a variety of examples of that. And so it's not hopeless in some sense. I mean, it's really a nice field to think about. But it's also incredibly messy. It's incredibly complex. It's full of surprises. And in some sense, it can be daunting, right? You'd like to describe something that can be described by one or two equations, if possible. You'd like to think about something that works in a predictable way. However, biology seems to be, perhaps by virtue of the fact that it relies on chemistry and very, very small objects, it's not as simple as perhaps we would like it to be. So when we apply mathematics, and I think at this particular, you can see that I was hoping to be very opinionated, and then I chickened out at the very end and say, in my humble opinion, here are the two questions that I think might be particularly important for mathematical biology to think about. From the standpoint of the other fields, what would cell biologists look for? What would other biologists look for when they talk to a mathematician or a mathematical biologist? And I think a couple of questions that I would think about as being truly important is trying to understand systems that are not fully characterized yet. So we don't wait for biochemistry to fully characterize some process before we model it. The process can be partially known, partially characterized. We may not know all the constants, chemical constants. We may not even know if a molecule exists. It can be a molecule X, a ghost molecule that is waiting to be discovered. But we can figure out mathematically whether it's likely to be there, and if it is there, what properties it may have. Another thing is to recognize that biology is an information science. It's one of those fields where information is the key, and one of the simple reasons for that is that we all carry information in our DNA. This is a storage of information molecule. It's unique in that sense. We also are exposed to the environment, we as cells, we as organisms, and the environment changes, and the key to our survival is to figure out how the changes in environment, all this information, can be perceived and then married in some way, or confused or suffused with the information that we contain in our genes. So a lot of that is about decision making, about information processing, and how to figure it out and couple that to the biological function, I think, is one of the grand challenges for us right now. Okay. So how do the cells do that? And this is a very famous slide, particularly for a biology community, a cell biology community. This is actually dates back about 15 years now, but it still is sort of a nice representation of the variety of networks dedicated solely to processing information, conducting information. And if you really squint, you may see in red color that many of those molecules, if they are malfunctioning, that can lead to cancer or some other really bad diseases. So understanding that is very interesting from the standpoint of how cells make decisions, how they process information. But also, if you figure out some ways this is done, you also may figure out how this can go wrong. And I think it's one of the themes of this conference, how things in biology, how the changes that may not be appropriate may lead to disease and ultimately may shorten our lives. So thinking about that, it's a wiring diagram, it's a lot of connections, a lot of errors, but what those errors represent, again, it might be useful to think about analogies with other communication information processing devices, and it has long been known, even for devices that are really well designed, and we think that we can design them almost in a perfect way, that they're always subject to this uncertainty, this noise. It may come from in physical devices from quantum mechanics, from thermal noise. In biology, it seems that we also subject the processes for information transfer are subject to noise as well. And this noise is not in your measurement. This noise appears to be intrinsic to the system itself, and it affects how the system functions and affects the outcomes. This uncertainty may be important. In fact, evolutionary theory assumes that there should be some uncertainty that can be selected upon, but it also means that when you describe systems like that, you need to be aware of noise and uncertainties. In fact, if you can, and as Ilya mentioned, in fact, with his help, we've tried to evaluate the capacity, information capacity of many of the signaling pathways, and the surprising fact was that it's very low capacity. It's about 2 bits that can be, at any given time, transmitted by many of the signaling pathways, something that a lot of people didn't believe before they measured that themselves. There are many, many papers now confirming this finding, which basically tells you that, again, it's messy, it's not very precise, or it's pretty messy collection of reactions. Nevertheless, using that messy information processing, we are capable of survival, life, and success. Right? And so how does it happen? How do biological systems use limited information about the environmental changes to successfully execute their functions? So that's one of the things that I think might be interesting to think about. And so what I'd like to suggest today is that if we have time, we can look at a couple of interesting examples. One other thing, as I mentioned, thinking about the organization of the networks. How do they should they be organized to execute certain functions? It's a very interesting question. Is the organization accidental? What if these networks, as they are, in fact, in biological systems, are there to execute multiple functions based on the same input? So if you have a signal, the information can flow to different decision centers, different execution ways, and how are the systems organized to achieve that? And so here, what I'd like to, if we have time, illustrate is that there are a couple of ways this can happen, at least. One is that the signals can diverge along different pathways and then converge onto the same target. And having a few different pathways actually is very useful for many different reasons. Or they can diverge and impact different outcomes, right? And in this case, the question is, how is it that we decide which of the outcomes is executed? Because the same input feeds into both of those outcomes. If the outcomes are mutually exclusive, it becomes even more of an issue. So what I'd like to talk about is a couple of interesting cases. One is related to decision-making, choosing between two alternative outcomes, and the other is related to how the cells sense the fields of external cues when they try to orient and move somewhere. And of course, before I talk about a really interesting physiological system, something that relates to us and cancer, I'd like to mention that, of course, we frequently would look at model systems, and one of the ones that I would again encourage everybody to revisit is Dictyostelium discoidum. This is a system that is interesting simply because it can exist as single cells or an organism. And so if you think about how is it that complex organisms may have evolved, this is an interesting example where cells, if they're well-fed, they exist as single cells, they don't really care about collective behavior. If they're starved, they come together, they form a slug, an organism that seems to be very simple but capable of movement somewhere looking for food. If that fails, they form a mushroom, and the spores from the mushroom fly away and colonize new areas. So it's a very interesting collective behavior as well as very interesting single-cell behavior. When that happens, of course, for that to happen, cells need to figure out how to come together to form an organism, how to converge on this spot. This is a very interesting process in and of itself. Of course, you can start seeing all this interesting spiral movement of the cells. They somehow can detect the centers of attraction. They come together. And a lot of the work has been, in fact, done to try to understand that. And so one of the things, of course, is that they need to figure out where to go. They exist in the waves of the chemoattractant that come from a single center. That center is not predetermined. It forms in a self-organized way. And in fact, multiple centers can form, and as I'll mention in a second. And so this collection of cells becomes an important and very interesting thing to look at. You can see that, as I mentioned, the signal comes from that center in the form of multiple waves, and those waves are maintained by the cells as they propagate. They contribute to those waves by undergoing oscillations, persistent oscillations of secretion of this cue, which turns out to be a small nucleotide CMP. And if you keep looking at how it happens, you find, as in the upper row there, that where the darkness basically represents the density of the cell, that transiently you can have a couple of foci or three foci, but over time one of them wins normally. So the cells, there are two different places that they can go to initially, and then one of those places is chosen over the other. When you think about how that happens, it turns out that the way it happens is that the focus that wins always has this magical frequency at which the waves are sent out. This is this magical frequency at which it happens. And the frequencies that are higher or lower lose. So the foci for those frequencies. And you can study that in a variety of different ways, but basically what it tells you is that the cell somehow can figure out where to go, but also measure frequency of the waves that approach and carry that information, right? So they need to measure gradients somehow of those cues, but measure gradients within waves, right? And so think about a wave that hits you. You need to be careful measuring gradients because gradients change. On the one side of the wave, gradient points one direction. On the other side of the wave, it points in the opposite direction. As the crest passes you, right? Think about concentration changes. The concentration increases and then decreases. So if you're trying to orient yourself, you need to do it in the right points of time. You need to, therefore, measure the frequency also to determine at which time points to measure the gradients. If you look at the actual circuits, biochemical circuits, again, everything is complicated. The circuit in a very simplified form is shown on the left of this diagram. But what you can do is use mathematical biology to try to understand how the circuit essentially works, how it can be in some sense reduced to something that is simpler. And that's something that we've tried to do a while ago and proposed one of the ways to do that, working also with Peter Divriotis, who really suggested that in a very qualitative fashion, but very inspiring way. And one of the reasons that we think about what has become known as LEGI, local excitation global inhibition circuit, is that the network, whatever it is, it has a few very important properties, one of which is adaptation. So if you think about two different pathways that initially converge and then diverge and then converge on the same target, if they can balance each other, you can achieve a transient response if one of the pathways is faster than the other. But then ultimately, if they balance each other precisely, the response can go up and then return back to the baseline. This is what's known as a perfect adaptation. So with perfect adaptation, it turns out that this circuit can achieve, but not many other circuits can achieve that. So and these cells that I described, dictyostevium cells, do have that property. So we say, okay, one possibility, for example, is that whatever the network is, it has to have that structure. And the reason we know that it has this adaptive structure is that we can use experimental tools, as shown in the movie that you can see at the bottom, to see how the network gets activated. In this case, it's just the translocation or increase in fluorescence of the membrane indicating activity of the response element, the final target. All right, so gradient sensing circuits turned out to be adaptive in much broader sense in many different other systems. So you start thinking, well, what is it about adaptation? How can we explain adaptation? There was this amazing story published a while ago in Cell, essentially trying different ways adaptation can be achieved and finding just two. The one that I just showed you, which is also known as the incoherent feedforward loop, LEGI, basically in this sense, but also negative feedback, not very surprising, can achieve perfect adaptation. So there are two ways of doing that, it appears. I don't think there's any mathematical theorem to prove that, so please prove that for me. But there are two ways to do that. One is negative feedback, the other is this feedforward loop. In fact, negative feedback is known to operate in another gradient sensing system that is quite famous, bacterial chemotaxis, right? And in bacterial chemotaxis, as shown on the right, you have slightly different architecture. So you have two different architectures that are adaptive and are used in two different gradient sensing circuits, okay? Why is that? Is it completely accidental, or is it for a purpose, if there is any way one potentially can be more beneficial than the other? Right, and so biology frequently would be saying that it's probably an accident of evolution, something happens, it gets fixed because it's useful, and you can end up with one circuit or another, right? Possibly true, but again, can we use mathematical biology to see what properties may be different about those two circuits, and perhaps one of those properties may discriminate and make one more useful than another in a particular set of circumstances. And so again, here what we can think about is what is it dynamically that's different about the circuits? And it turns out that they're very similar, not only in terms of adaptation, but by comparing them, even though in some sense it's comparing apples and oranges, but you can sort of cross-normalize them. You see that both of them adapt to step increases in the concentration. Both of them can respond to pulses of input. And in fact, both of them turn out to be bandpass filters, meaning that they respond maximally to the optimal frequency, the magical frequency that I mentioned before. So there's no way to tell them apart in that sense. They also can perform, both of them, what is known as the fold response, sort of try to figure out how the signals are presented to them, and so on. There's one way, though, they're very different. And one way they're different is in this middle row, and this is where you stimulate the cells not with just step inputs or oscillatory inputs, but with a continuously increasing input, a ramp stimulation. When that happens, and of course you can do it with different rates, right? The ramps can be of different rates. It turns out that under very general assumptions, the feedforward loop, the LEGI circuit, will be able to adapt to that, whereas negative feedback circuit will not be able to do that. And the levels to which the negative feedback circuit will settle, right? It's not going to be the baseline. It's not the initial conditions, but different levels, and those levels will be proportional to the rate at which the ramp is presented, right? Whereas for the feedforward loop, what will change is the peak and to some degree the position, the amplitude and some degree of position of the peak. Okay, and you can show that in dictyostelium, which we've done a while ago, that indeed these cells do adapt to ramps, whereas it has been shown extensively for many other negative feedback loops, including bacterial chemotaxis, that they don't. The negative feedback loops don't adapt to ramps. So that's curious, interesting. How can that be useful? Again, let's revisit the idea of how dictyostelium cells sense gradients. I already mentioned that they sense gradients within waves that hit them. Right, so it's like being on these boats, and you can be on two different sides of the wave. So what you would like to do is to try to orient yourself, see where the concentration is higher. You can do that. You're a big cell. You can tell the difference between front and the back. But you're doing this as the waves are passing, and so the concentration goes up and down. You don't want to be affected by that. Right, by the fact that the concentrations go up and down, unless, of course, you're trying to measure that, again, in temporal way. You're trying to measure the changes in time. But these cells don't seem to be doing that. They're doing the spatial sensing, the sensing of the difference between front and back. So they would like to get rid of this extra information in some sense because it can interfere with that sensing. And so they would like to adapt to the transient increase and do the spatial sensing in this sense. Now think about bacterial cells. Bacterial cells are different. They are small. They cannot do the spatial sensing. The only way they can measure gradients is by moving from one point to another, and therefore if they adapt to the ramp stimulations, they are losing precisely the information that they are interested in, and therefore doing that in a way where they don't adapt would be a much better idea. So there is a way for you to discriminate between the properties of these two circuits and find a way, one which may actually, for a specific set of conditions, may be preferable versus the other. Again, because in one case you would like to get rid of the changes in time. In the other case, it's a crucial information. It turns out that for Legge circuit in Dicty, it's a little bit embarrassing. We actually don't know the identity of those two pathways to this day. And yet everybody believes that, and we have ample experimental support for this model. There's been a flurry of papers recently, including one from our lab, where experimentally we show with beyond reasonable doubt that it's a Legge model. Again, this is where mathematical biology is incredibly powerful. We don't know the circuits themselves. We know what they have to be for the system to work. And they're just waiting to be discovered. In some sense, it's like physics, where a lot of predictions have been made, and then experimentalists finally find a planet that has to be there, or a particle that has to be there. So I'd like to argue that in biology we can have a very similar situation. Like, for example, knowing exactly what biochemical properties of the circuits are based on a combination of modeling and experiment. All right. So a small conclusion for this part, and we'll see if do we have time? Yeah. I think we have. So there is a difference between these two different adaptive systems, negative feedback system and the feedforward system, both of which are adaptive and have very similar properties all around. There's still nevertheless difference in certain ways they perform that could be evolutionarily advantageous. Now, let me switch gears, but retain this interest in gradient sensing, but also go back to decision making. And what I'm going to show you is something that relates to blood vessel formation initially in the fish, but I'll show you, of course, that it's also true for how blood vessels form and reform when you have normal development of humans, and most interestingly, cancer formation, where a phenomenon known as angiogenesis, growth of new blood vessels to feed the tumor, happens. So in zebrafish, this is this nice fish animals, you see the gradual development of the organism in a very nice way. They're transparent, so you can see everything basically in the microscope. And when you look at the developing vasculature, blood vessels, they're all there for you to see. And so what I'm going to show you is the movie at the bottom, and then I'll amplify it a little bit. You'll see those blood vessels actually growing, but you also will see that they're flashing. And the flashes you'll see in live organism are calcium flashes, flashes of the increases in concentration of calcium. So if you look at this, what you should be looking at is this area over here. You can see that there are this faintly, maybe you can see that there is an appearance of this long sprouts, right, blood vessels that are extending. Let me try to show you this in a hopefully amplified way. So again, where those numbers were, you can see that there are these flashes, and gradually you may be able to see the extension of blood vessels. So why is this interesting? Maybe it's completely accidental. It turns out that calcium signaling is critically important for a fundamental decision in this case, whether the cells divide or move. They cannot do this at the same time. They either divide or they move. The selection between these two different behaviors, or as biologists would say, phenotypes, is a crucial one, of course, because you don't want to enter into one and then do the other, right, at the same time. And so for a growing blood vessel, which basically schematically this would look perhaps more clearly than in the movie, would look like this. We have an existing blood vessel, as would be, for example, in a cancer. There's a cancer cell that needs more oxygen. Somehow there's a signal that is presented to the blood vessel cells in the form of a growth factor called VEGF. And that triggers a whole series of events, including the growth of new blood vessels towards those cells. And some leading cells will have to move, and the cells behind them have to divide to fill the trail, right, to be there in the wake and allow the growth of the blood vessel. All right, so again, you can go into the signaling network and find it to be very complex. If you reduce the complexity just a little bit to focus on what happens based on calcium in this system, you'll see that there are, again, two different, or in fact more decisions, but two of them relate to either proliferation or migration. The migration decision is less well understood. The proliferation decision has been linked to a factor called NFAT. It's a transcription factor. And we may want to just look into this just a little bit more in detail. So what we find is that when the cells are exposed to different concentrations of growth factors, of this growth factor, we have calcium essentially responding in three different words. It has language that consists of three different words, right? One of them is this response where there is a single profound peak as a function of time, and then non-zero, it doesn't return to the baseline and stays relatively high above the baseline, although it's very hard to see there, but it is different. Another word in the vocabulary of calcium is spiky oscillations that appear to be quite stochastic. And finally, no response, but it's a very reasonable, important word. So at different levels of stimulation, different concentrations of growth factor, you will elicit those three words on the level of calcium, in the calcium vocabulary. And what changes with different concentrations is the selection of the frequency of these words. So as the signal is very low, there's primarily no response, and to some degree there's some single peak SP or random spikes, but you can see that the frequencies of those responses will change. The properties of responses themselves do not seem to depend, as I'll show you in a second if you have time, on the concentration. It's just the selection of those three words that depends on it. And you can show that if you look at two different responses here, one is average NFAT, and you can do the same thing with the rate of cell division. You'll see that what happens is that only the cells with high level of NFAT will be also exhibiting both the division property, increased division property, but also enrichment in this random spiking behavior, right? So you'll see that on the left-hand side, you see the high average NFAT only happens alongside with these repeated spikes. On the other hand, the average motility depends, it seems, on this single peak and this persistent signaling of calcium. So there is a almost deterministic separation of behaviors here, depending on whether the cells display a single peak, in which case they may go into the increased motility, or they have repeated spikes, in which case they may increase the rate of cell division. All right, so a single input somehow in a vocabulary consisting of three different words somehow is translated in a very robust decision-making, although still subject to some noise and stochasticity. And one way to explain how this can be done robustly is to think about such properties of these pathways as memory, or if you are a physicist, you may be inclined to thinking about ratchets, right? And ratchets could be of two different types, either a nice ratchet that you kick it and it goes forward but never comes back, or a ratchet can go back and forth, back and forth. So each if you think about steady input of calcium, where you have single peak and then a steady input, will push forward both of those ratchets to some degree, but we'll keep even this ineffective ratchet activated continuously. Whereas if you have a spike input, the imperfect ratchet will go back and forth. Perfect ratchet will ultimately progress and accumulate the activity, integrate the activity over time. So with that, and one important assumption that the activity, whatever the processing memory and the inputs are, it has to exceed a certain threshold. You can actually show that this simple idea, the difference in memory, can allow you to discriminate between these different inputs. So when you have the single peak in the persistent response, that will primarily activate the systems that, or exceed the threshold, the systems with imperfect ratcheting, while imperfect ratcheting will not be able to integrate over time and consistently at all times exceed the threshold of the response, and vice versa. So because of the lack of time, I'm not going to lead you through a very detailed process of how we've convinced ourselves that this model in fact is true. Maybe show you a couple of glimpses. Of course, NFAT is something that has memory. It can integrate signals over time. You can show that it works in this system. In fact, let me show you this movie. Again, I like movies, and this movie is quite rich. It shows you how experimentally you can look into that, where you have 3 different outputs in a single cell, one of which basically shows you the calcium output as at the baseline. Yellow color, if you see it, you'll see some pulses, represents increases, and fat is a green color, a red color, I'm sorry, and it will translocate the nucleus in one cell and will stay away from the nucleus in another cell. You also see a cell dividing, the blob divides. So you see that in the dividing cell, for example, if you look at it, there's this continuous oscillatory event, and initially the sister cells will be synchronized, which is interesting. But more importantly, look at cell 1 and cell 2. Cell 1 accumulates NFAT in the nucleus, and that actually is also correlated with the oscillatory calcium signaling in that cell. Cell 2 doesn't have oscillatory signaling, and the accumulation is very transient. So if you focus on the cell here, you see that there is a transient translocation, and then the signal goes away. So with these types of imaging experiments, you can really try to see how calcium, again, is translated in the probability of cell division through NFAT signaling or increased motility through being able to stay above a baseline. So let me just say one more word about this without, again, bothering you with some of the experimental details. Again, you can perturb calcium in a variety of different ways. For example, if you're interested in studying how this low but meaningful increase of calcium above a baseline, whether indeed it's critically important for increased motility, you can inhibit calcium or just pay attention to the cells which retain high level of calcium versus the cells that don't. And you can see dramatic differences in the motility output. We also can show that have shown that the calcium sensor in the case of migration, enhanced migration, is a molecule called MLCK, myosin light chain kinase. So what do you do with this information? Let me just say a couple of words about that. What you do is you can design a model which has a few components, one of which, as I mentioned, VGF, this input, can allow you to select between 3 different words. In this case, it's shown as a W waveform. It also can control some of the other things I didn't mention too much, but for example, in the repeated spice, you can think about frequency. In the steady output, you can think about how long the steady output lasts. I told you that if you interrupt it, the cells will not move as fast. In fact, the critical threshold has to be exceeded for both of them to generate the output, both the NFAT translocation and cell division and the increased motility. So how do you figure, the connections between this? Is it that VGF determines both the selection of waveforms and some of the properties of those waveforms, or just the selection of waveforms? And again, it turns out we can consider probabilistic models in this case, Bayesian models, and you can select the model that is shown in red, in the middle model, as the most likely model, or the model that provides you with the outcome without the complexity, the extra complexity that you don't need. And with that model, you can take that model and you can explain something else. What you can explain is not only how the cells make decisions whether to divide or to move, but also you can predict what properties the directed cell migration, the chemotactic cell migration will have. And so again, it's a very noisy process. It's a super stochastic process that has certain deterministic features. And nevertheless, you can predict how the cells will respond in different concentrations of VEGF. And you can do experiments to test that in microfluidic devices, which actually allow you to test the prediction of the model, stochastic model, with the experimental observations, which will be noisy, but you can compare the outline of the predictions on the right-hand side with the experimental data that are embedded as dots within that prediction field. All right. So let me not take this further, but rather spend the time I don't have to say one more thing. Ilya has been very kind to me, and I'd like to stop myself for a change. I'd like to just suggest strongly that mathematical modeling can be very useful if tightly integrated with experiments for a couple of reasons. Let me just tell you again what I think might be those reasons. One of which is when we have something happening and you can really test it by, as a small child, by shaking the box in all sorts of different ways. You can infer what the properties of this unknown circuit, even though the circuit may be actually appear to be very complex, what those properties essentially may be, at least for, or important for the process that you're trying to understand. And mathematical biology can be very powerful in prediction, having this predictive power of what has to be in such cases. It also can be very important in unraveling very complex processes of decision making and continuous processing of complex signals that are presented to the cell. And a variety of different mathematical models can be used, from ordinary differential equations to probabilistic models to just essentially arguments about different architectures of the networks. So with that, I'd like to encourage you to think about those questions, talk to me about the new Institute of Systems Biology at Yale that I'm heading, and have a good time at this conference. Thank you very much.
Now we have experiments to follow and try to find those pathways. That's nice, but I always wondered whether there are any pathways actually existing. I always felt like it's always networks. A pathway is just that we want to separate a particular set of genes or molecules that they're interacting, but it's never a pathway. It's always a network. So how robust is your predictions that it's just these two network based on adaptation or some kind of, I could imagine that if I take more components into account or some more complexity, I will come up with many more predictions. And so how robust is this prediction of pathways, right?
I got it. So it really depends on your question. Or a quest your own, right? People have different quests. For example, somebody is very excited about a certain signal. It could be VEGF that I showed, this molecule, or TNF, which is a signal that we also looked at. And your quest could be, well, what is it that this particular input can stimulate? And it can stimulate a lot of different things. And of course, you'll see that a lot of different processes are regulated through very complex intermediate processing. And this will be most definitely a network question, not a pathway question. There are really very few, if any, cases, and mostly probably in bacterial cells, where you see one-to-one connection, meaning that there may be some intermediates, right, some chain of events, but there's going to be some input and some output in a very definitive way. Having said that, your question can be a little bit different. For example, if you think about how this single input translated through this single intermediary, in this case calcium, can result in making alternative decisions. For example, and again, you cannot make both. So the big question here is that having some calcium signal, right, you'd like to process in such a way that you can have an unambiguous, with this noisy situation, unambiguous decision whether you may increase cell proliferation or cell motility, but not both at the same time. So for that, it turns out that what I showed as two branches, right, I mean, that's a network view, right? It's just not a highly interconnected in the middle network. It turns out that such decisions, not only in this case, but we have many, many more examples. If you're interested, I can talk to you about this, where this idea of some common regulator and then two branches, each one leading to two different decisions, in fact, sometimes mutually exclusive decisions, can proceed without any extra crosstalk or cross-activation between them or cross-regulation between them. It's a very surprising situation. We are still trying to understand how this happens, but it seems to be prevalent in biological systems. And so the question of how is it that they decode in an unambiguous way the input is a very important question that we try to address here. So yeah, biology is complicated. It's all networks. It really, in some sense, depends on what you're trying to understand. If you're trying to understand what VEGF can do, you cannot deal with just this simplified system. You have to describe everything. If you're trying to understand how VEGF controls cell migration versus cell division, you can have a simpler view of this, which works. Yeah.
So the examples you showed included oscillatory inputs. Are those special in a sense in these decision-making networks?
So oscillatory inputs are interesting in two different ways. In fact, I mentioned two different situations, one of which is where they're decoded by something that is a bandpass filter. In this case, there's an optimal frequency that the cells would respond to. That's why they select one of the foci to go to. They can be maximally activated by a certain frequency, and that's the magical frequency that I mentioned at the very beginning. The other case is where you have perhaps a simpler circuit, a circuit that is not bandpass filter, it can be a low-pass filter, but has high enough memory or good enough memory. In this case, frequency of the oscillations can be very important to see whether the output is going to be achieved. So yes, they're interesting, but I asked a question maybe perhaps in the second part in a slightly more general way. If you have something like that, how do you make sure that you don't do something else? Right? That you would have one pathway with reasonable memory not being activated by some other inputs, for example, constant level of the input. Yeah.
Let's thank you. Let's thank Andrei again.